Cells are the building blocks of life. In our research, we've identified that disease cells look differently than healthy cells in many contexts. We developed a technology called image-based profiling to study cell morphology. We develop open source software for the image-based profiling technology. This allows us to analyze large-scale screening data sets that use either genetic perturbations or drugs to identify different reagents that might make disease cells look healthy again. Our open source tools include Cytotable, Cosmic QC, Cytomining, and Buscar. Although all of these software tools contribute to the overall mission of image-based profiling and drug discovery, we decided to separate out each tool into an individual package. This is because each tool is complex enough to justify its separate development, and also to make sure that each can be maintained by an individual developer. We identify what we're supposed to do, and then we put it, just actively working on it, and then it's done. So my lab develops methods and software for analyzing microscopy images of cells. We do this mostly in the context of something called phenotypic drug discovery, which screens thousands of different drug perturbations on cells to make cells that have disease look like healthy cells. In this context, a perturbation is anything that we might want to do to perturb the cells in some way. So this could be a genetic perturbation or a drug perturbation. It just changes the cell in some meaningful way that we can measure with image-based profiling. We do this also in the context of various specific diseases. So we focus on neurofibromatosis type 1, NF1, pediatric cancers, all different types, and cardiac fibrosis. The tooling that we build, the open source software, is important for us because we use it every day, but we also want the whole community to benefit from our open source tooling.
This looks important. I work on Cytotable. My main role there is as a research software engineer to help make sure that the data get processed really quickly and to make sure that I'm using best practices to help maintain that software over time. Cytotable is a Python package that helps to prepare data for analysis by combining images and image feature data into one consistent format. This helps researchers spend less time wrangling data and doing custom coding and more time focused on their biological research. Cytotable outputs can then be used in Cytomining to perform further analysis. Another innovation that Cytotable offers is the ability to output data in either AND-data or Parquet files. Parquet files were one of the first formats that we introduced into Cytotable because of its ability to be high performance. And when we're dealing with a lot of data like this, we need any data that we access later to be high performance. Parquet is special because it's a columnar table type, which is different than, say, an Excel spreadsheet file, in that you can access individual columns without having to open up the full contents of the table. It's also high performance in the sense that the data are always the same type. Every time you read a Parquet file, that data that's kept inside that file is read the exact same way into memory based on the reader, because it's all stored in what's called Apache Arrow data types. These types give you consistency across multiple different programming languages, which means that no matter if you're writing in Python, R, Julia, Rust, you always get the same data out of that same file, which means that for scientific work that we get consistency and reproducibility, which is a major advantage that we have through Cytotable.
For example, this is literally this is the cell segmentation here, and you can see that there are multiple different...
Cosmic QC is a software package designed for quality control of single-cell morphology profiles. During cell segmentation, technical artifacts, debris, and erroneous segments can arise, which can lead to issues during our analysis. These issues can occur from many different sources. These can include microscope issues, contamination from dust or unwanted material, which can include maybe coughing on your samples, which sometimes happens in the lab. Cosmic QC helps to label or flag incorrect segmentations, which you can then filter from your downstream analysis, which supports more accurate and cleaner results. So we know that if we have an object and the segmentation is going off of the object, we know that that is incorrect. We want clean, smooth lines around that object instead of it going off and haywire. So, for example, people with their iPhones can actually segment things and make stickers, but it's easier for a person to do it on one photo because they just click and hold the one photo and it'll segment and outline it. In our case, it is not as simple because we need to do that across hundreds of cells in one image. So we need more complex and sophisticated algorithms to do that.
How many drugs are you able to test at like a time or in like an interval of time?
I mean, so our initial screen was 550, but in our lab we tested over 10,000 probably.
So Cytomining performs the image-based profiling pipeline. So what it's going to do is it's going to take in features that we've already extracted from microscopy images and process them through an image-based profiling pipeline. So that means it's going to annotate the single cell, so it's going to tell me which treatment the cell came from, which well. It's going to normalize the single cells, so it's going to make sure that all the features are on the same scale. And it's going to perform feature selection, and feature selection is just to make sure that all the features are informative in the profile that we then analyze downstream. Cytomining takes as input cytotable formatted data, and so cytotable takes in different features from different tools. So this is things like Cell Profiler or Deep Profiler or other proprietary formats. And from there harmonizes it into a format that gets it ready for Cytomining. So Cytomining basically takes in a Parquet file and then performs the image-based profiling pipeline and then outputs a series of Parquet files that we can then analyze downstream. Cytomining also performs something called batch effect correction. And so batch effects are technical artifacts that might arise from technical differences across different plates and different batches. And so Cytomining has algorithms that would perform batch effect correction.
Like SCAR.
Yeah,
SCAR.
they would look like what you would imagine SCAR look like.
A lot of my projects revolved in developing methods for image-based profiling, specifically for, in my case, high-content screening. And so one of the softwares I developed was Buscar, which is a play of words of to look for, which is in Spanish and Portuguese. And the idea behind Buscar is that it allows researchers to find or to search for a compound that is suitable against a specific disease in a high contact manner. When you, let's say, you have a population of cells, right? And then within that population of cells, you would have some subpopulations that are different, going through a different biological process. But when you take an average summary behind that, you are condensing that into a single representation. So you're hiding all the biology that is associated with that population. And it should be very important to account for when doing high content drug screens. So Buscar avoids the whole summarization part and treats that whole population as a whole at a single cell level. So now we have the biological nuances that were never included in current metrics, which uses basically average populations. So what Buscar does now is it finds drug candidates at a single cell level, which to my knowledge, no metric does that. And I feel like it gives you a more accurate perspective on how a compound interacts with the cellular population.
Our suite of open source tools supports the field of image-based profiling. It is our hope that image-based profiling and the work of our software can help to narrow the funnel of drug discovery so that we can discover new therapeutics for diseases of unmet need.